So, very warm welcome, everyone, to this Cosmos seminar event. It's our pleasure today to host Paola Castaño, University of Exeter. She worked with Sabina Leonelli on a very interesting topic. She'll talk about experimental practices and data workflows in plant-space biology. Thank you very much, Paola, for being here today. I leave it to you.
Thank you, Sylvia. Thank you very much for the invitation, and thanks everyone for joining. So yeah, I'm going to give you sort of a brief overview about where I'm coming from with my approach to plant-space science. And then I'm going to focus particularly in the field in which I have conducted research most recently, which is plant-space biology. Sorry, I'm just going to set my timer because I don't want to, yeah. And then I'm going to talk about the experimental program and some dimensions of data practices that are my entire argument here is to try to put these two aspects into in close relationship with one another. And finally, I'm going to close with some broad questions about the future of the field as the experimental program moves from low Earth orbit towards the moon. So a few years ago, I embarked on a project to understand scientific research on the International Space Station. I started with the puzzle of what this platform even is, because it makes a lot of promises. It's a multi-purpose endeavor, one that has been also quite controversial and put into question by many different actors. What's the purpose? It's a multi-purpose scientific facility, which in and of itself means that it has to spread itself very thinly across many different scientific domains, and that poses a number of questions about what kind of scientific practices take place there. But also, of course, there's the geopolitics of a space station, of an international space station as an endeavor. So next to these big promises and this kind of multi-purposeful nature of the platform, I'm very curious about the intricacies of experimentation, about and the fragility in many ways of experimentation in a domain like this. And this is one thing that, in a way, has fascinated me throughout this research. And finally, I'm interested in the assessments of value around the space station. I mean, broadly speaking, the station is surrounded by critics and supporters, and each one with various types of involvement and with expectations and with entitlements, kind of selectively select aspects of the space station to make their case about why it is a very important endeavor and has benefits for humanity versus perspectives that mean basically that precisely it tries to do too much. It doesn't do a few things well enough, particularly for this community, the 1993 episode by which the superconductor super collider was at the table of discussion in the U.S. Congress, and the space station was at the same time being, very in this kind of battle of justification that took place there. This is an interesting moment when particularly these communities in the fields of astrophysics and physics have been the most kind of vocal critics of the space station as a scientific endeavor. So of course, one question as a sociologist, I mean, it might seem very naive to just simply, is it a laboratory? Well, not entirely, right? It's more a complex data collection environment, and I'm going to zoom in some key aspects of that complexity in what follows here. What kind of platform for scientific research is it? And I'm going to give you a very, very technical answer, which is a tricky one. I'm going to, of course, give you more detail about the trickiness of the endeavor, but that I think is a key starting point to try to understand what scientific practice might look like there. And as a sociologist, of course, how can one approach that? I mean, for a long time, this was a joke amongst my colleagues. I'm an ethnographer, which, ethnography by definition means participant observation. How come you're studying scientific practice in a place where you cannot physically go? But of course, the process of these experiments, has multiple locations and it's actually spread across many different actors. And I think that's a sociological task to really trace that systematically. So my approach is to follow the process of conducting experiments and what I call the various social worlds in which they participate. And there are three types so this notion of the social lives of experiments, basically I break it down into the study of an experiment on the space station entails taking very seriously this whole realm of promise, this realm of what the aims are, what the large pictures, what the horizons of the achievable are once an experiment is proposed, the intricacy of the operations, but last and not least, the very different assessments of value around space station experiments. So I selected three experiments for the book that I'm completing. One in plant-space biology, which is a veggie experiment in which astronauts grew and ate lettuce on the space station. So this is kind of the plant science experiment, and the one that really got me in this community and this line of research, the one that I will talk about in more detail later. The twin study, which is a multi-omics investigation, the first of its kind, where an astronaut went to space for approximately a year, and his twin brother stayed on the ground with 10 different teams, studying them in a very systematic and multi-omics way, which was the first time that NASA sort of ventured into these fields, which also opened up a whole set of ethical questions about genetic data for astronauts, decisions, counseling about potential findings. So this is a study that took place in 2015. And then finally, one that perhaps is closest to the interests of you all, which is the alpha magnetic spectrometer, hosted by CERN, and as a particle detector, basically a cosmic rays particle detector, but also sent to the space station under the promise of detecting dark matter, and even antimatter. In fact, the A back in the days, the alpha stands for what the space station was called at some point, the Alpha space station. I mean, the space station has had its own complicated history, but in principle, it was called the antimatter magnetic spectrometer. So these are the three experiments that I've looked at and that I've examined in this very close way. So I've been to all these places. I've been to Kennedy Space Center, Plant Science Labs. I've been to big conferences. I've been to rocket launches. I've done a lot of archival work at NASA headquarters and the European Space Agency archives. I've looked at operations. I've been to CERN, to the mission control of the alpha magnetic spectrometer. And in all of these various social worlds, I've tried to piece together this understanding of what the social life of an experiment on the space station is. So when you look at it from a distance, you see this notion of, yeah, this is an experiment. When you break it down, you start thinking about the various moving pieces and the three kind of sets of actors on which I've focused when it comes to the execution of experiments are, of course, the researchers themselves, the operations teams, and the astronauts. What is expected from the side of a space agency, from the researcher side, is that they downscale. That is, that they minimize to the largest extent possible the technical requirements, the size, the crew time required, the telemetry. Make your experiment as small as possible to fit the multiplicity of activities that I have to take on the space station. Then later they have to delegate, in many cases, crucial data collection tasks, at least in the case of the first two experiments, the life sciences experiments. When it comes to the AMS, except for servicing, in fact, this happens it's separate from astronaut activities on the space station. So this delegation from the researcher side is made to people who are highly skilled multitaskers. That is, they are not experts, of course, in the many specific fields in which they have to conduct experiments. And then finally, deferral is vitally wait a long time till your samples come back and the analysis can be conducted. The operations team try precisely to compartmentalize the hardware and the task. They translate this know-how of researchers in their labs into very detailed tasks that astronauts receive. And then they distribute, of course, the tasks also when it comes to the management of samples. Astronauts handle fixed experiments in some cases. They execute these tasks. Just to give you an example, procedure on the space station can involve, things like, Velcro, the tool to, a surface in order, in most labs, of course, the notion of the tool flying away from the experimenter is not a concern. Well, in this context, it is. And things even of that sort are detailed in the instructions that the astronauts get. And when it comes to analysis, astronauts are not, with very few exceptions, they're not involved. So I've thought about this in terms of this notion of modularity. I mean, of course, modularity is a characteristic of the space station. It is the material infrastructure of the space station. The space station is made of modules that were progressively attached to each other, starting in 1998 with the Zarya module from Russia, docking with the United States Unity module. But modularity, from my standpoint, is not just this physical property or even like a deliberate organizational strategy, but it is the process by which heterogeneous elements, modules, very much physical modules, equipment, frameworks of justifications, meanings, and practices sort of contingently dock with one another. So modularization, basically, I see it as this kind of I have this processual sociological perspective that in which I see all these pieces in constant movement. You have movable pieces of different sides, no core organizing principle. There's not necessarily a hierarchy between elements, and there's always, of course, the constant possibility of compartmentalizing or connecting elements. So this kind of physical structure of the space station also serves for me as an analytic to understand the nature of science as I described it above, with all these moving pieces between the researchers, the operations teams, and the astronauts. So I always take a step back and ask, why does it matter? And particularly for you in the fields that you study, well, of course, space science is commonly associated and evaluated in terms of astronomy, astrophysics, heliophysics, and planetary science. I mean, only in 2020, the NASA biology program became part of the NASA Science Mission Directorate. Before, it was only in the context of human spaceflight. And of course, once it enters that domain, it becomes evaluated under the terms of these other fields. And at least I hope I've conveyed so far that it's a completely different type of scientific endeavor. It also matters because it also puts into question these notions, these kind of popular notions of discovery, and I call them as ta-da moments or like events, which of course for which, I mean, particle physics, or at least in the kind of more common imagination, or, the image of a black hole, the detection of gravitational waves. Like I always put these points of reference as examples of what the space station hasn't achieved and perhaps will not achieve. Because particularly when we're talking about biology, what you have is the incremental understandings of systems and processes under very complicated conditions that I'm about, again, to describe in more detail. And finally, this matters because systematically identifying enablers and obstacles for research and about public perceptions of science continues to matter more every day, as we need to inform broader publics about what the fragility of the scientific endeavor is about. And I do like that word, and plans in particular bring that into I think a closer territory when it comes to what my most recent research has been about. So plants in space are. Sorry, I'm just going to check the chat just to see if there are any questions. Oh, I think it's just people that have had to leave. Okay. So plants in space are kind of fascinating in the sense that they offer a unique window into to the tension between the big promises and the more intricate and fragile operations. When you look at these images of like greenhouses against the arid backdrops of the moon and Mars, where you see astronauts contemplating chilies or fresh vegetable or a flower floating amidst the equipment of the space, these have become somehow familiar images. Of course, the first two, the top two, from left to right are renderings that but the other four images are actual photos taken aboard the International Space Station, and they have become increasingly common when it comes to the role of plants in space. Astronauts frequently talk about these experiments in their public appearances. They're very photogenic experiments as well. Of course, the color, the green contrasting with the wires and all the equipment on the space station. But a much less familiar of plants in space involve tiny Arabidopsis thaliana seedlings in a petri dish in an aluminum disc. Part of this that becomes part of, but it's actually called BRIC, and it does look like a brick, but the BRIC is also an acronym for Biological Research in Canisters, with a petri dish fixation unit. That definitely looks and sounds less sexy than the beautiful greenhouses on the surface of planetary surfaces. But in fact, this has been the core of the research program in plant-space biology, where scientists have studied Arabidopsis thaliana, the model organism for plant science in general, an organism whose genome has been widely annotated and through a very, very fascinating process of international scientific cooperation has become the model organism for this field, and a hardware that is developed by NASA in the 1990s, that it's also in many ways a story of the ingenuity that involved in taking experiments to the space station. Astronauts cannot handle fluids, they cannot handle chemical fixators for tissues once they grow in space. So they've had to develop a system like this, where through this kind of caulking gun that looks like it, astronauts basically open compartments here in order for liquids to enter, both to start the experiment, in which for the most part the seedlings grow in the dark, but also to stop the growth of the tissue at the given end of the experiment. There's your perhaps for some of you, known Italian astronaut Samantha Cristoforetti manipulating one of these units in what has been, as researchers call it, their workhorse. These bricks have basically been the core of the experimental program. So when you think about what space is as an environment for plant space biology, well, it's this complicated thing that the space station is. And I break this, and I'm going to go back to this image when we talk about data, because this is basically a timeline of the process of one of these brick experiments where days before launch, but also, of course, months before that, in the actual scientist laboratories, all the preparations begin to send these petri dishes, these brick units, basically, to the space station. The sterilization, all these protocols have to be very detailed, right? The seeds are irradiated, then they're stored in the dark in a cold unit, then they're assembled into the petri dish, then the little reservoirs are filled, then they're inserted into the bricks, then they're put in these larger containers, then they're mounted into the vehicle, into the cargo vehicle. And then what you would think most traditionally as an experiment in which you have a control and you have an experimental group, well, here the story begins to get more complicated. Once the experiment is launched, astronauts remove the bags and they inject they simply allow they start the experiments by allowing the plants to, the seedlings in this case, to grow for 8 days. Then they inject this chemical basically to fixate the tissues to stop the growth of the plants. Then they're loaded into a freezer. And then they return to Earth by splashing onto, in this case, the Pacific Ocean. I have a paper in collaboration with the ISS Archaeology Project in which we trace what happens with these samples when they return and the effort to really keep temperature as stable as possible. But of course, just by looking at the top part, you will see a lot of fluctuations when it comes to the temperature to which these samples are exposed. Meanwhile, on the ground, at the Kennedy Space Center in their facilities, they have this chamber that in principle aims to mimic all the environmental factors of the space station, but of course it cannot really mimic all of them, except for temperature and CO. The ground experiment, the ground control, sorry, starts at least 24 hours after the flight samples begin. And then finally, you have the analysis process when both flight samples and ground samples reunite. And again, there's various sorts of manipulation that take place. And I'll go back to this specific graph in a moment. So this is basically in a snapshot, of course, decades of plant-space science that I've just tried to summarize for you. So I'm just checking my time. Okay. So now what happens when you look at this at scale? And when you look at it at scale in terms of comparing data sets, this was the story, the very intricate story, highly summarized, of one brick experiment. But what happens when you put many experiments of this sort next to each other? Then this requires us to take a step back and understand what open science means and has meant for more broadly space biology. And this is work that I conducted as part of the philosophy of open science for diverse research environments that Sylvia just mentioned. It's a project, an ERC project led by Sabina Leonelli at the University of Exeter, where I worked with her, and now the team moved to the Technical University of Munich. There are several case studies, and my case study was about this data repository. So basically, it's the social life of experiments now in the context of data sharing practices. So I studied this repository called GeneLab, which came online in 2014 in response to the 2011 decadal survey for the National Academies of Science in the United States, basically aiming to maximize the scientific return and utilization of the ISS. When you look at the saga of the operational saga that I've described, of course, there's a question of, well, this data cannot simply be brought back and stay in the scientists' labs because this has been federally funded, and this is a result also of a broader movement, at least before 2025 in the United States, about open science and open access to federally funded science. So GeneLab is very much NASA's response to that in the domain of space biology. So it is a database, a specimen repository, and a collaboration space for omics data from biological investigations in space. So apologies about the typo there. And it aims to enable, and this is the key phrase, crosstalk amongst valuable experimental biology resources. That's basically what is stated in their strategic plan. But what's fascinating sociologically here about this is, I mean, this could have very well been an archive, right? Scientists finish their experiment, they send their data, end of the story. But precisely because of the complexity of the data and the need to have as many eyes as possible examining this data, the repository managers in 2018 came up with the idea of having analysis working groups, which are basically groups of experts that are interestingly, and this is another very interesting finding of this study that I cannot talk about in detail here now, but it basically expanded the space biology community by bringing people not only who have been directly involved in the execution of those experiments, but external eyes, a lot of external experts with an interest in omics. And this, of course, not only refers to plants, but it also refers to rodent experiments, to microbe experiments. Human data is not part of Gene Lab, although there have been a couple of initiatives, I mean, separate from core kind of NASA astronauts, but through more private initiatives where that have enabled controlled access to human data. Because of course, as you can imagine, this is quite a delicate matter. But these analysis working groups have been a key actor in this context, and they have two roles. One is to provide feedback to the repository on scientific standards for the reuse of the data. But also, of course, to collaborate to reuse the data to conduct analysis. So from 2022 to 2025, I conducted an ethnographic study of the plants analysis working group. I became really embedded in this community. I attended approximately 80 meetings. I've interviewed people, and of course, I followed, which is the key outcome of at least the first 2 years of operation of this group, which is this paper called Meta-Analysis of the Spaceflight and Microgravity Response of the Arabidopsis plant transcriptome. When you look at the word meta-analysis in the context of the life sciences, is quite a common practice, increasingly common practice. In this domain, this is the first one of its kind. And we will at least hope to give you a sense as well of why and some of the complications when it comes to this. The key finding here is that secondary data analysis, relying on metadata workflows, which I will define in a second, is providing novel ways to interpret, compare, and potentially integrate results obtained in these experiments. These experiments are highly heterogeneous, so the protocols that I described in terms of temperature management, in terms of seedlings management, etc., of the timeline of these experiments vary a lot. Basically because these are experiments that are allocated as a result of a call from NASA. So different labs have different criteria, different specific questions. Some researchers are more interested in particular molecular responses to gravitropism. Others are more interested in, I mean, scientists have different scientific questions. They have different protocols. So it's a highly heterogeneous landscape. So if you look at the image below, of the 15 data sets that they analyzed in this paper, in the gray boxes, you have all the various types of hardware used, and each hardware is its own story, the different types of vehicles used, the different types of tissues involved in the experiment, and of course, depending on the time in which they were done, because for the most part, these experiments are conducted using RNA-seq. Some of the older data sets use microarray, and not to get into details, but basically it's all about sort of the coverage and resolution of these analysis methods. So it's a very, very messy, heterogeneous landscape that these researchers found when they began to ask the question, what are the common biological responses that we find in plants across all these experiments converted into data sets that are found in the repository? The answer is, well, actually, these experiments cannot be directly compared to each other precisely because of this variability. So what the paper ended up becoming was more an analysis of the batch effects of the kind of confounders involved in attempting these comparisons, and zooming in again on experiments conducted within the brick hardware that use the same type of RNA-seq. So basically, after identifying these confounders between the 15 data sets, they ended up with 2 data sets that basically shared that method of analysis on the hardware, which also involved the sharing of lighting conditions for the samples. So what we found as a key and really interesting scientific contribution of the paper is what we in a paper that I wrote with Sabina Leonelli, we call metadata workflows, which is basically the effort, which, the first 2 points seem like pretty typical sort of data curation practice. You annotate the characteristics of data provenance, you select and frame metadata to suit specific analytical and interpretive goals. But then it gets more analytical, right? When you begin to categorize metadata either as background variables, potential confounders, or relevant experimental factors. And you begin to use statistical methods to analyze metadata as relevant factors for viable comparisons between data from these experiments. So this is precisely what led to zooming in the two particular experiments in which they were able to capture signals that in fact had not been possible to capture in previous isolated experiments, such as the effects of markers of oxidative stress, particularly regarding the plant's sort of difficulties. Hypoxia effects, basically, lack of access to oxygen in a way. And in a way, that's also something that is induced by the hardware. So again, what is the environment here? It's a layered, um, the space flight as an environment for plants is a layered domain, basically. So in this paper that we published last year with Sabina, we first provided this account of the operational constraints, the heterogeneous experimental designs, and the partially unknown environmental factors that make up this experimental domain. And this is very important because it again frames or characterizes the novelty of this practice in these specific fields. And we make an argument about how we acknowledge that experimental control are the central epistemic challenge in plant-based biology. And metadata workflows, in fact, epistemically function as an ex post type of experimental control, precisely aiming to disentangle the various factors involved in the experiments and trying to trace them to the biological effects on the plant. So, textbook definition of control is you have a group that has received an intervention and a group that hasn't. All other conditions are kept the same for both groups. Well, this is, of course, the idealized version of what a control is. What I showed you when I described this story here is, well, there are aspects of experimental design that are under the researcher's control in the green square. The red square, you have the operational constraints to which they have to sort of submit their experiments, and there's this overlap between experimental design and operational constraints is meaningful there because again is a scientist maneuvering to get the most out of the limited possibilities they're given when they send their experiments. Then you have this big issue in the purple square of the unknown environmental factors. Temperature, yes, I mean more or less, but if you look at the thermometer, you have a lot of fluctuations that are very difficult also to mimic on the ground. And this effort on the ground facility to again mimic as many possible aspects of what's going on in space. And then finally, you of course have in red, you continue to have the operational constraints in terms of all the various efforts to transport, to keep the samples at a particular temperature. And then you have the scientists in their labs conducting their analysis. So removing the picture of the actual concreteness of the experiment, what we have is that a sample is precisely at the intersection, is kind of all these many different aspects, right? I mean, environmental factors, some of which are known and unknown throughout the process. And experimental design, which is a set of principal choices that researchers make when it comes to designing their experiments, the operational constraints to which they have to subject, and then finally, the analysis method that they have chosen. So again, why does the study of these data workflows matter? First, because in this sharing of data and in the detailed curation of metadata, as I've described and as we described in the paper with Sabina, space moves at least from being this kind of intractable cluster of stressors towards a more differentiated group of experimental factors, and that's what the paper written by the analysis working group achieves. So this work, and we argue here, that can be regarded as an extension of experimental practice because it restructures the empirical resources on which the researchers build their analysis, the analysis of past experiments, but also can contribute to the design of future ones. So this is, yes, retrospective reconstruction, but then this is also perspective building because in the groups, what you see as a result of this engagement with data and this possibility of access to all these various data sets is that there's an effort to design better controls for future experiments in what will very likely be a much more complex scenario of even more reduced capabilities when it comes to scientific experimentation on the moon. And now, very briefly, I want to talk about where this field is going and how they're envisioning what's next in terms of what has been learned. Was there a question on the chat? Oh, I can't see it now. Anyway, I'll get back to it in a second. about to finish. So in 2024, at the European Low Gravity Research Association, the core community of plant-based scientists got together for a workshop precisely aiming to discuss what has been learned and what's the future of the field. And perhaps expectedly, the issue of standardization has become quite salient. And this is something that, again, data sharing has had a lot to do with that. I attended the event, and I'm a co-author on this paper that came out last year that first provided this updated collective vision for the future of plants in space that presents an overview, particularly the last decade. And why the last decade? Because the previous paper of this kind was published in 2012, again, as a result of a workshop at the same conference where, I guess fewer people at the time got together and wrote something similar to what this paper is, which is it's a viewpoint paper. So this overview focuses on the last decade, particularly showing the advances in omics investigations like the one I described, and in the study of gravitropism, which is the response to plants to gravity as a stressor or as a cue. Again, the key of the conversation, the center of the conversation was the need for standardization, the effort of international collaboration, which of course this was written at the end of 2024. Published in 2025, but the workshop took place in 2024. That is an increasingly complex scenario, considering, of course, the funding, the volatility of the funding conditions for science in general in the United States at the moment, but also for NASA within that context. And also, of course, the establishment of shared priorities. And one of those priorities tackles the question of what is required to move this research program to the moon. And this is an effort at scaling up, basically. So what you have is fundamental science, moving from fundamental science to crop production with these aims of, helping astronauts, supplement their diet, eventually pharmaceuticals, but also the larger aim is what they call bioregenerative life support systems, where plants can help to recycle air and keep sort of what has been an aim for a long time, the closed loop in the habitats where astronauts and other organisms will coexist. So the issue of scale here is fascinating because, of course, we have the epistemic aspect of scale, right? All biological experiments, I would say, all experimentation involves a challenge of moving between reduction and amplification, right? Scientists break down a complex problem of interest into a set of manageable parts for a particular set of devices and methods, and then they make larger inferences about the larger composite problem. So here, they introduced the bioregenerative life support system readiness level. We're basically going from 1 to 9. These are the steps that the scientific program has to fulfill in order to be able to actually have plants as a functional part of these life support systems. But in my perspective as a social scientist participating in these conversations, scaling up is not just this epistemic problem of inference, it's also a move towards much more explicitness when it comes to the value commitments and the institutional embeddedness of this scientific program in our current geopolitical scenario. So I'm proud to say that my two contributions to this paper, and I apologize, there's something with the image, are priorities number 10 and 11, in which open science has to be part of the conversation. And they were very receptive with that, basically building on the experience that I've described before with the NASA repository. And the question about whether moon exploration is even acceptable for many people in the world at this point. And I said it perhaps too bluntly, but I continue to think this is an image that, that is very salient for our times, is that the flags that will be planted on the moon in the next Artemis mission or in future human missions to the moon are going to also come alongside the drills that will be part of extractive economies around the moon. And there's an open question about how these scientists themselves look at this scientific program that has all these various moving pieces, all these various unsolved puzzles, and all these various, tasks to achieve, how they see those aspects and how they view their research as part of this larger picture. So moving to the moon, and this is my last slide. There are some questions that I hope I can spend the next few years, answering in collaboration and in this dialogue that I think it's a unique dialogue that I've been able to establish with this community, but also one that I really a conversation that I really want to have with them. And I understand that as any deep conversation, and I'm sure you've had this with your interlocutors, with your scientist interlocutors, sometimes gets to uncomfortable spaces. Here is perhaps the most simple question, what does it mean to move from the cabin ecology of the space station to the moon as a more complex ecosystem? What does that mean for experimental design? The key question about what it actually means to talk about sustainability when, it's a word that gets thrown around a lot in this context. How does the organization of this research, combining academia, industry, and space agencies from different countries, can shape different processes and outcomes? How this kind of New York, because this is a new scenario, the first funded plant experiment for the moon was the kind of grant was from NASA was allocated to a private company in Arizona that they are collaborating with university researchers and with space agencies, but this was allocated to a commercial actor with, of course, plant expertise that led them to win the grant, the funding. But it is a new configuration of actors that is institutionally poses a lot of new questions as well. What are plant scientists' own ethical questions about human habitation in space in relation to their practice and their perspectives about environmental implications of human activities on the moon? And how can the scientific community foster responsible research practices? We've heard a lot about responsible research in many domains of science in the last few years. Will this be a useful framework to think about here? I think it's something that is worth trying and testing, and particularly because it is a framework that is quite open and quite conducive to dialogue and to constructive dialogue with the scientists conducting this research. These are sort of where I'm taking this trajectory of interest on plant space biology towards the future, and that's, as I said, what I hope can occupy my time in the next few years. And well, I've been very lucky to be funded by all these various institutions to conduct the research, and I'm happy to hear your questions and to start the conversation.
Thank you. Thank you very much, Paola, for this very, very interesting talk. So we can now start the Q&A. So I ask the participants to raise their hands in the chat. Their questions.
Sorry, I cannot see how I can erase.
There's reactions
Yeah, and
or
yeah, but then it says reaction settings.
interesting. Okay. While I solve this
Okay.
issue, you can ask your question.
Okay.
Go ahead.
Thank you, Sylvia. Yeah. Yeah, thank you, Paula. That was really fascinating. And I do have a question about this analysis working group that you mentioned, because it's very interesting. So, I think you said that, this kind of group is kind of thought to have some, it's in order to have some external eyes, from outside that can contribute in like giving feedbacks on scientific standards and so on and so forth. And you mentioned that there are, for instance, like people working on microbiology. And so I'm very like excited to think that, a scientific community opens up to another scientific community and tell them about, scientific standards.
Mhm.
So I wanted to know more about this. And do you think that this is also a way to build up together the scientific standards of, a community?
Mhm.
And the people who are invited in this working groups, so do they have like very close scientific standards or, very different scientific standards? How do they choose? The kinds of researchers that they want to invite as constituting the analysis working group. I just want to hear more about this
Thank you.
structure.
Yeah, they are also like a fascinating kind of like they're hard to characterize in the sense that, of course, in particle physics, you have the collaboration, which is something that is a lot more structured, or you have more kind of organic networks. This is something that is both organic and orchestrated. So it
Okay.
started in 2018, as I mentioned, when the repository first asked two questions. We need some kind of like community accepted standards, but we also, who is going to use this data? Is this just going to like die basically on a website and no one's going to come and look at it? We need to make this repository alive, like a resource that is alive. So at the beginning, it was an invitation only kind of endeavor for people who had conducted the experiment to agree on an analysis pipeline. Because as I described, part of the picture of heterogeneity is all the various analysis methods um, for the experiment, basically for the data. But then it expanded into a much more open-ended endeavor in which they're open to anybody. People do have to fill out a
Hmm.
form and sort of
Oh,
describe their
wow.
expertise. But there are students, there have been citizen science projects that have come out of the analysis working group. So we're talking now about a group that began in a workshop of about 25 people, now to something that is 1,300 people who participate in these groups across all these various. So in principle, there was one for each organism. So you had animals, microbes, plants, like it was very much just that. And now you have female repro. So for instance, an OBG, like a gynecologist, a specialist, a practitioner, a clinician who lives in Florida. And so all the rocket launches said, wait a minute, what's happening with standards for female health when it comes to this? She contacted NASA, contacted the analysis working groups, had access to some data sets, in fact, ended up having access to some astronaut data. The repository, of course, has a code of conduct, has some way to like also kind of govern the groups. There's a charter, but you see this combination of someone just being interested, of course, with
Yeah.
expertise, but not that expertise having
Yeah.
nothing to do with spaceflight, joining the group and producing, a paper that, for instance, showed signals of like showed, I mean, not of course through the actual study of the hormones, but through liver signals, issues related to insulin and estrogen in mice and in female astronauts, based on this kind of brokered access to the data sets. So anyway, that's just like one snippet of a story, but basically the point here is that this has expanded the scientific community by bringing all these non-spaceflight experts into the domain, particularly in the context of the plants group, in which I have observed the interactions a little bit more. What's really interesting, of course, from the outside of the space biology domain experts is they ask about controls.
Hmm.
Like, wait a minute, what's the ground control in one of these experiments? How come the conditions on the ground are not exactly mimicked? How come you don't have all the telemetry from an experiment? How come you don't have the vibration? How come you don't have specific radiation dosimeters attached to the hardware? And the reply from the scientists doing the experiments is like, we don't. Is the nature of the beast over here? Is how we manage to do, is this downscaling basically that I
Okay.
described.
Yeah.
So in that conversation, I think both parts learn, of course, the kind of non-domain experts learn about the particularities and the complexities of this domain, but also the ones who have maneuvered in these operational constraints keep asking how we can do this better. How can at least we agree on a set of parameters for the experiments in order for them to be comparable at the end of the day? Because what they've found so far is precisely these challenges of the experiments not being directly comparable. So I will share in a moment a link. I conducted a survey of members of the analysis working groups where I kind of detail a little bit more what the group's compositions are, because a lot of the people, again, I mean, at least half of the respondents had not been involved with spaceflight experiments before, and that's a huge thing in terms of
Oh,
bringing a bunch of new people to the field. Maybe I'll just stop there and we
yeah.
can
Thank
move
you.
on.
Thank you
Thank you
very
for the
much.
question. Yeah, I'll post the link in a sec.
Thank you.
Yeah.
Um, yeah, Valeria.
Mhm. Yeah, I just wondered, you mentioned the paper in History and Philosophy of Science, so I was wondering, was it from a historical perspective or was there philosophical problematics, and if the latter, what was the philosophical part? Because what you were telling seemed to me mostly sociology, and I think you even mentioned sociology at some point. So could you explain how you position yourself with respect to different domains and with respect to philosophy in particular?
Thank you. Of course, yeah. So the paper was part of a project that is a philosophy of science project, and a project that is also more broadly part of the philosophy of science in practice, sort of sort of subfield, I would say, within philosophy of science, which is open to dialogue with the methods of the social sciences. I'm a sociologist. I'm trained in sociology. I'm not necessarily an STS scholar, although, of course, when you say sociology of science, a lot of people associate that. I'm more a sociologist in the sense that I study institutions and knowledge making and more from a kind of like, Chicago school type of sociology, which is where I was trained. So that's sort of about me. The paper is both. I mean, I think the paper is three parts. I mean, history, because it is the first paper of plants about plant-based biology in this journal. I mean, I just posted the link that does tell the history of these complexities, at least not in detail, but it does provide an account of what experimentation on the space station looks like. And there's always this historical consciousness about writing this, because as I tell my interlocutors, the scientists in this field, I'm writing their history in a way which hasn't been written anywhere else. I mean, this is not a field that, of course, has had a lot of interest from social scientists. I mean, astrobiology is a different story, where there's a lot more philosophical interest in it. But space biology, that is sending organisms, earthly organisms as we know them, out there to see what happens to them while they're there, has not really been that studied. So there is that historical component. The sociological method there is because, of course, of the nature of the observation, of the access I've had to the group, about the fact that I was able to follow the meetings, the discussions about the paper as it was produced. So it's kind of like, it's a way of following a scientific practice, not a lab practice, but a data workflow practice. And then finally, the argument, it is a philosophical argument. I mean, we are in dialogue with the literature on experimental controls in philosophy of science. So we basically, and more broadly also, like philosophy of data in the life sciences. So we precisely what we're trying to do is bring together the experimental and the data workflows aspect. So that in itself is because a lot of people talk about the access to large volumes of data really means that some aspects of biology are becoming post-experimental. That is an argument that has been made in some parts of philosophy of biology. Well, the question here is that this is very much part of the experimental program. I mean, it's not just ex post data analysis. This is part of a program that is alive, that is ongoing, and that is still trying to figure a lot of things out. And this has had an impact in the experimental program. But also, we're making the argument that metadata can actually serve the epistemic function of an experimental control in the ex post analysis of data, not necessarily in the actual execution of the experiment. And that's an argument that we make in the paper, a more philosophical argument that we make in the paper.
Yeah, thank you for details. You want Sylvia to
Yeah, I have a follow-up actually, because I would like to learn more about the very last point that you mentioned, Paula. Because of course, it seems to me that when we talk about experiments in space, there are very much, I mean, there are very different constraints from the experiments that we perform on Earth, of course, on the labs. And of course, one of the main characteristics when we philosophers of science look at scientific practices, when we look at scientific reasoning, we assume, in a certain sense, that experimental setups can be, that developed in a way that assume or presume reproducible protocols. We generally this holds for physics. I mean, we do a huge amount of cross checks. That, however, in space are less, I mean, we can't do that in the normal way, let's say. So what are the strategies to, if any, of course, that also relate to what you just mentioned in your reply to Valeria? So how do we have to look at epistemological questions that are pivotal for the philosophy of science when confronting with these important practices, because I think that what we are going to witness in the next decades will be precisely described by your work. I mean, when talking about experiments in space, even in the 70s or in the 80s, when there was the Cold War, and at some point. There was the detent and scientists, both Soviets and the Americans, were performing joint experiments in space, or at least they interacted on biology experiments. So there weren't physics experiments in orbit. So I leave it to you.
So, yeah, of course, there was one thing that we talked a lot about with Sabina when we started, writing this paper, and even when you look at the kind of philosopher that has worked mostly, that has worked the most on experimental controls in the life sciences, Jutta Shikoe, who's a um, I mean, basically, she says even in the most idealized, notion of the world, no experiment has the perfect control. No experiment can achieve perfect control. Of course, this is an extreme case of that, right? So there's two choices. One, you evaluate it against the standard and said, as many people have said, this is really not good science. This is substandard science. And I mean, why engage in these very expensive experiments? Our approach to that is non-evaluative. We in fact look at the fact that this is an ongoing scientific practice, that it is taking place and that is changing, and let's look at how scientists deal with these complexities. So the first is the survival approach, which is the one that I think has been the one that predated the data sharing approach, which is each one does what they can with their experiment. I think the decisions are principled. I don't think any of these scientists will just simply say that they felt like the petri dish had to be put at a certain temperature. Like, there are protocols that are established in their labs and that they have followed. By Giovanni, that they have followed. So, I mean, and this is what they translate to space. So that I think that in and of itself, you can look at it as scientists basically maneuvering in a complex system in the best possible way. And that's always a fascinating thing for the study of science, how they manage to do that. So that's one moment of the question. The second moment is precisely when this experimental program meets the data sharing, which is something that it was not envisioned. I mean, these data sets were not envisioned to be shared. So at the beginning, there's this apprehension of your colleagues are going to look at your experiment. I mean, just to give you a very basic example, some of these petri dishes, in some cases, they put 30 seeds to germinate on the space station. In others, they put 300. In others, and in the 300 one, they have to put a lot of sugar, like a kind of sugar, a kind of nutrient, basically, to get the plants to grow under this kind of insane conditions, absolute stress, versus in the other one, they need low sugar. That in itself is a confounder that makes these two experiments, even if everything else was comparable, very, very hard to compare. So what the data sharing program is doing is that it's making the scientists enter a dialogue with each other about the nature of these choices. And that is epistemically valuable. But what's interesting here is that this has happened not through the experimental program, but through the data sharing program. So in many ways, there's a lot more infrastructure knowledge, as I call it in something that I'm writing right now, generated as a result of the exposed data sharing. Again, with these retrospective aspect of integrating to generate insights, but also prospective aspects, how we can move towards more standardized experiments. And then the last point, in answer to your question, is precisely events like the one I described at the European Low Gravity Research Association in a Viewpoint paper, like the one that was published at the end of last year, in which I was able to collaborate. And it was also very fascinating, as my contribution on these two specific points was being made, to see precisely the discussion around standardization. It is a must. We have very few opportunities. And interestingly, the experiment that is about to go, I mean, that is about in 2 years' time, but the next Artemis mission, which is this planned chamber that is going to be located on the surface of the moon, one of the investigators was the person who wrote the Gene Lab strategic plan. So he's someone who's very aware of data sharing. This is an experiment that is being conducted under the premise that data will be shared and the standards are transparent for the community and they will align as much as possible to agreed upon sort of so the PI of that project is a co-author in this new phytologist paper, in this viewpoint paper. So these people are in a very active dialogue with each other about precisely how to improve that. And I feel it's just fascinating that I'm kind of witnessing this in real time, as they move towards a much more standardized program. And is any effort of standardization, and it's true of any field, there are choices being made. And there are choices that will be put into question by the next generation. You know, what someone's noise right now, what someone's background noise right now could be the future signal for someone. I mean, I think in your domain is a lot more, that metaphor flies a little bit further than it does in mine, but it is in a way the same, right? Is making the data as transparent as possible for future uses and making every decision that is built into a data set. Also, this is why this notion of metadata workflows is important. It's not just simply annotating the data, it's precisely what is generating the conditions to extract biological insights from already existing data, but also to design future better conditions for data production. So, yeah.
Yeah, thank you very much.
Sure.
I don't know if there are other questions. No? Okay, then we thank our speaker. It was
Thank
really
you.
intriguing. I learned a lot.
Thank
And this was, for me, I was very much looking forward to your talk. It was very informative and very stimulating. I'm sure that we will talk more about these topics. And I also would like to remind that tomorrow there is another Cosmos seminar, which I would say is very much related to your talk, because it will explore the problem of property right in space law, because this is
you.
the hot topic, let's
Yeah.
say, nowadays, and it's a topic that nicely, I mean, there is we can bridge in a sense the gap between the philosophy of science and, or at least the philosophy of science in practice and philosophy of law.
Mhm.
Because as we shall see tomorrow, the big challenge today is to rethink of property rights to ground international space law, and so I mean, not only philosophers of law, but also lawyers are looking for interaction with philosophers because they have to conceptualize in a different way the property right on the face of space science development and future missions. So thank you again, Paola, and
Thank
hope
you.
to see some of you also tomorrow for
Yeah, I shall be there. Okay. Thank you,
Thank
everyone.
you.
Thank you.
Sylvia, bye.
Thank you all.
Thank
Bye
you.
bye. Bye. Bye.